Okay, guys, we are going to get in and get out of chapter 6. It's very short, it's just on a little bit of metabolism, how processes happen in your body. So, if we get into it, guys, your book starts off with talking about some different organisms that use bioluminescence. You ever seen the movie Finding Nemo? There is an anglerfish that also has a little dangler on its front of its head, I guess you could say. And it emits a bioluminescence, like a lure for fish. So the fish go, oh, look, pretty light. And then all of a sudden, boom, it's eaten, it's gone. So you can see the squid here, it has bioluminescence. This is just part of its, it's a regular squid there, part of its characteristics and part of its body. But anyway, so all the chemical reactions that happen in your body contribute to your metabolism. With the squid, it had some type of chemical reaction that emitted a bioluminescence, so it could, shine that little light. But anyway, so we're going to go through some of the different chemical reactions that happen in your body. There are different metabolic pathways. We're going to go over 2 of them. But each one, we're going to use different enzymes to catalyze these reactions. So usually what happens, guys, is there is more than one enzyme in our whole reaction, okay? Last chapter, we went over the primary messengers and secondary messengers. And with respect to that, we had a cascade of different ligases being used in order to get to that end activation. It's kind of like the same thing in enzymes. Originally, it was one enzyme, one reaction, but we're finding now that it's actually more than one enzyme for a reaction for the whole thing to take place. Now, each specific reaction, the reaction is very specific to one enzyme, but usually it's more than one enzyme to get to that end product. So there's catabolic pathways and there's anabolic pathways. The first one is a catabolic. If you think of catabolism as our word for that one, we are breaking things down. I don't know why I always associated it with this, but I always think of catabolism and cannibalism. They sound the same. Not that I'm condoning cannibalism, but if you are a cannibal, you're breaking an organism, in that example, a human, down into smaller pieces. Kind of weird, but yeah, catabolic reactions, they break things down. The opposite, anabolic, this is building up, making things bigger. So if we went back to the macromolecules, we had the dehydration condensation reactions and the hydrolysis reactions. The dehydration condensation reactions went given monomers and you're building it up to make a polymer. Okay, that's going to be an anabolic pathway. That's going to be an anabolic reaction. The opposite of that, hydrolysis, was when we had a long polymer and we're breaking off monomers. So that one's going to be catabolic. So different forms of energy here. We got kinetic energy, that's in motion. Thermal energy, it's still kinetic energy, but it's associated with movement of atoms or molecules. Whenever these atoms or molecules move, guys, we give some type of heat off, and that's thermal energy. That's why it's really hard to get to absolute zero, negative 273 Kelvin, because why we can't get there is once we get really, really, really cold, our atoms start to move just a little bit. And when they move just a little bit, that is heat generated. Okay, so they start to warm up again. And then lastly, potential energy. This is the energy based on a location. So the organism isn't necessarily moving, but it's in a location where it has the potential to maybe perform some type of energy or the potential to move. And then lastly, chemical energy. It's just for chemical reactions. Okay, it's still a form of potential energy. Okay, so this is kind of what is stored in a chemical structure for chemical energy. So like lipids and things like that, they would have a lot of chemical energy because we can break all those bonds. All those bonds are like potential energy, and they can all release energy when broken. So here's a little picture from your book, guys. All right. The person all the way up top here, they have a whole lot of potential energy because, they have the potential to move down. The people in the water have a lot less potential energy because they're already at, I guess, the ground floor. Although if they drowned, I guess you could still consider that potential energy. They'd be moving down, but anyway. All right. So thermodynamics, study of energy transformations. We are going to have an isolated system, and we can also have an open system. Isolated systems, the surroundings do not matter. Open systems, we can have an energy transfer from those surrounding areas. So there's different laws of thermodynamics. I know you guys have probably heard of these in 7th grade science, or if you took physics already. But first law, energy cannot be created or destroyed, but it can be transferred. So in other words, guys, we're never going to create energy, but we will transform it. We're going to move that energy. We're going to use the energy. And even when we use energy, yeah, maybe we lost energy, but maybe that energy went to something else and it absorbed our energy. So energy is never created or destroyed. So that's our first law. The second law, I'll come back to that picture there, is every energy transfer increases the entropy of the universe. Now, what is entropy? Well, entropy is a measure of disorder. So in other words, guys, if everything was in one little ball, everything is very organized in one area, all right, we would have not a whole lot of entropy. But as we start to take things away, think about like your bedroom. If we are going to just throw things all about your bedroom and not clean anything up, your bedroom, we just increase its entropy. We increase its disorder, okay? So if we go back to here, our first law, yeah, the fish has some sort of energy, and it lost that energy, and the energy went to the bear, okay? The second law is with the bear, it ate that fish, it used some of that energy, and as it's using the energy, some of that energy leaves its body. It might sweat some out. It's breathing carbon dioxide out of its body. So we're increasing that entropy in the universe. Right? Okay. We're going to skip that one. All right, I'm just skipping through some of the stuff we don't need. So as we have entropy increase in one area, or I should say decrease, in one area, like you guys, we can have the entropy decrease in your body, but as that happens, something else is increasing. So it's something like mass not being created or destroyed, just like energy not being created or destroyed, okay? So entropy is the same way. If it increases in one area, it's going to decrease in something else. It's cause and effect. Okay, so we're going to go over a few things of free energy. We're going to make it super quick. So free energy, what free energy is energy that can do work in something living. Okay, so we have a certain amount of free energy that we can use. How to calculate this is it's the change of G, okay? So we are going to do our final state, in other words, our end energy, minus our initial energy in order to calculate our change of G. Give me 1 second, guys. Lights went out. Sorry, that's really annoying there. Okay, so some of these are going to need energy as an input for this reaction to occur. Okay, so some of them are going to need energy, some of them are not. If we get an end product, so a change of G, that is negative, that means it's spontaneous. That means it happens without energy being needed by the system. So whenever we have these spontaneous reactions, we call them exergonic. What this is they are actually going to release energy instead of needing energy. So our change of G is going to be negative. How we tell this from now this is a bar graph. You can use a line graph to tell this as well. But up here, our reactants have a higher energy than our products. So in other words, if we drew a line between these two bars, it would go down. That means it is exergonic. Okay. The opposite is endergonic. This is one that is going to need energy to occur. Our change of G is not going to be negative. On this side, in this example, it's going to be positive. And this is where our reactants are lower than our products. Okay. So we already talked about this, guys. Endergonic ones, they are not going to occur naturally. They're not going to be spontaneous. In other words, we're going to need energy in order for the reaction to occur. Okay, last one here. So energy coupling is going to be an exergonic reaction being used to drive an endergonic one. In other words, exergonic gives off energy, endergonic absorbs energy. So if we use them both together, we're kind of using that energy given off by the exergonic to fuel the endergonic. So we talked about ATP. ATP is our molecule we use for energy. It's fueled by the breakdown of glucose. It's made out of 3 parts, and we really didn't go over these parts when we originally went over ATP. But we are going to have adenine, which is a nitrogen base. That is the difference between GTP and ATP. GTP is going to use guanine instead of adenine. Everything else is the same. We're going to have a sugar, and that sugar is the same sugar that is in RNA, which is going to be ribose, and we're going to have 3 phosphates attached to it. Now, whenever ATP gets converted to ADP, we're obviously losing a phosphate. Here is our structure of ATP, and we have it going through dephosphorylation. So we have 3 phosphates here. Whenever it becomes dephosphorylated, we now only have 2 phosphates down below. 1 phosphate is no longer attached. Whenever that bond is broken between the second and the third phosphate, that is when we have a release of energy. Okay, so this is a hydrolysis reaction. Whenever that bond is broken, guys, we already talked about energy is released. We talked about it being a hydrolysis reaction. So I think we're good on these ones. Just make sure you know, guys, the phosphate is that broken off phosphate is not what gives the energy. The broken off phosphate, that bond breaking, is what releases the energy, not the phosphate, because later on we're going to reattach that phosphate. Okay, catalysts and enzymes, we talked about these in the past. Catalysts, non-living, they're non-living things. They speed up the reaction without being consumed. In living things, we call them enzymes. They're going to reduce the amount of energy we need for a reaction. Okay. Here is an example of the breakdown of sucrose. And we talked about this before. Sucrose is a disaccharide. It's made out of 2 monosaccharides, glucose and fructose. When we break down this disaccharide and go through this hydrolysis reaction, yeah, we do get 2 monosaccharides out of it. Yeah, we do need water in the reactants to break that bond, but we also need an enzyme in order for this to occur, and that enzyme is sucrase. Yeah, it's pretty easy, right? It just ends in ASE. So we already talked about activation energy. This is the energy needed for a reaction to occur. Enzymes are going to lower that activation energy. So it's not that activation energy, if we're looking at a graph, is not going to be as high with an enzyme. Okay. All right. So here is a activation energy graph. You guys can see here. We have the reactants on the left. We have the products on the right. The reactants are above the products. Okay. So this is going to be an exergonic reaction where we are going to have a negative change of G. Okay, so we're giving off energy in this. But the EA in the middle here, that is our activation energy. If an enzyme were present, this hill would not be as high. So we still have the same amount of energy if we use an enzyme. The enzymes do not change that amount of energy. Okay, so here, the red line, energy present. The black line, the red line, enzyme is present. The black line, the enzyme is absent. All right, but still, the change of G would be the same in both of these. All right, going back to the substrate enzyme complex. You guys remember there's an active site on that enzyme, and that is where the substrate is going to bind to it. Now, we talked about a lock and a key model before, and that lock and key model just means that the substrate and the enzyme are very specific. We have one enzyme that's going to fit one substrate, and only that substrate. Now, the induced fit model was a little bit different. What this says is that as we have this enzyme, okay, the substrate might not fit perfectly, But whenever we have this enzyme, we might have different molecules come in order to help that fit be absolutely perfect, okay? That's the induced fit. So yeah, it might not fit perfectly, but with the bringing up of these molecules, the substrate will fit perfectly into it, okay? And there's your complex there, guys. You can see the substrate fits very nicely into the enzyme. The induced fit means, yeah, after it goes in, this side might close so it even fits better on all sides, okay? And then once everything's done, guys, we can keep reusing this enzyme, so it's like a cycle. Things that can affect an enzyme activity, in other words, its active site. Temperature and pH are the two main ones. We can even have salt levels, salt concentrations. They can affect it as well. But what happens here is if the temperature or the pH gets too far out of whack, yeah, the enzyme becomes denatured. It changes its active site. So we have an optimal temperature that each enzyme works at, and it's different for every enzyme. And we also have an optimal pH, okay, whether it's acidic, basic, neutral, where these enzymes work. Obviously, stomach ones would, want to work really well in a acidic environment. Here is pepsin and trypsin, okay, two different enzymes. This one, pepsin, okay, Pepto-Bismol settles down your stomach. Pepsin, it's an enzyme that works in your stomach, works really good at a pH of 2. Trypsin, it's in your intestines, which are slightly basic, so it works really well in a slightly basic environment. Take the trypsin, put it in the stomach, it's not going to work at all. Put the pepsin, put it in the small intestine, it's not going to work at all. All right. Last thing with enzymes is allosteric regulation. So let me get to the picture of allosteric regulation. What goes on here is we are going to have two types of regulation. We're going to have inhibition, which is our first one, and activation, which is the other one. So inhibition means something gets inhibited. So if we have an inhibitor and it binds to our enzyme, that's going to change the active site so the substrate can't bind, right? So if the inhibitor is bound to the enzyme, the substrate can't bind, okay? The opposite is going to be an activator. When the activator is bound, what that does is it changes the active site so the substrate can bind. So allosteric regulation, if it's an activator, the activator needs to be there, bound to the enzyme for the substrate to bind to it. The opposite, the inhibitor, if the inhibitor is bound, the active site changes so the substrate cannot bind to it. Okay? And then feedback inhibition, we might be forming something. This kind of goes with gene expression a little bit, but we form something in our pathway. And I'll show you the picture of it here. The end product of our pathway, and this one, an amino acid called isoleucine is being made. Once that isoleucine is made, and we do not need any more isoleucine, what we are going to do is take that isoleucine, we're going to go bind it to that enzyme, bind it to that protein. And when it is bound, that's going to change the active site so that whatever is being used to activate that enzyme can no longer bind. That substrate can no longer bind to it. So that's feedback inhibition. All right, guys, so that is chapter 6. I said it was going to be really short. I just need to give you a few things. We'll start chapter 7 tomorrow. Have a good day.